All right, good afternoon. Today we're going to be looking at the structure of DNA a little bit. Last time we watched the video on Rosalind Franklin and some of the controversy behind Watson and Crick's ultimate discovery that DNA's structure is relatively simple. Today we're going to be kind of using a model of DNA. So the first thing you're going to need to do is get some materials. If you're here in class, get a few materials off the counters. If you're watching this from home, you should remember us working on this in class with some construction paper and some scissors. But assuming that you have done this, then we're going to kind of explain some of the what the pieces are. Again, Watson and Crick, James Watson, Francis Crick, were given the credit for finding the discovery of DNA structure, in large part due to Rosalind Franklin's research, and that's maybe the one fault that I have with them, is that they should have given her some credit for that. The first thing that they discovered, due to her pictures, her X-ray diffraction pictures, was the signature of a double helix, which is kind of like an X pattern. So if you notice from the photo that they referred to in the movie, it had a distinctive X pattern, which meant that DNA was helical, and not just a single, but a double helix. And we'll refer back to that occasionally next week. The second major discovery that they put together was that DNA was made of some very simple building blocks that repeated over and over and over. These repeating subunits are called nucleotides. The structure of a nucleotide is relatively straightforward. There's three parts, as I've shown there on the right. One part is represented by just a letter P, one is a letter S, and the third component is something called a base. But this whole thing is called a nucleotide. So when we refer to that, think of these little three-part building blocks. All right, what are these three parts? One part is called a phosphate. We name it a phosphate because it has the element phosphorus in it, along with 4 oxygens. And so what this ends up looking like is kind of a pyramid. Here's a phosphorus, and you'd have 3 oxygens coming off of this, and then there would be an oxygen up at the top. So you'd end up with this kind of pyramid-looking object called a phosphate group. The S stands for sugar. This is a monosaccharide, or a pentose, because it has 5 carbons. And you should know the name of it. Deoxyribose is the sugar. And if you notice, the phosphate is on top of the sugar. That was the brown part. The sugar is the blue part. And then the third component, which sticks off into the middle, is called the base. And this has the element nitrogen in it. And this is where DNA gets its complexity. Nucleotides all have the same structure, but the base could be any of those 4. All right, what are the 4? These are ones you need to memorize. There's 2 basic categories. If we could group these 4 bases, there'd be 2 in one side, called the purines, and 2 in the pyrimidines. The first ones are called adenine, or sometimes we just write it the letter A. And its equivalent on the pyrimidine side is a thymine, which starts with the letter T. And what Watson and Crick kind of deduced was that A's and T's connect to each other in the middle. So the sugars and phosphates make up the sides or the rails, and these bases kind of stick out into the middle of the DNA molecule. The other two bases, guanine, G, and cytosine, C, these also connect to each other. And the reason we call them purines is because they have a shape that looks like that. The pyrimidines have a shape that looks like that. And you do need to know which ones are the pyrimidines and which ones are the purines, and there's a easy way to tell the difference. Pyrimidine has the letter Y in it. So does thymine and cytosine. The other two bases, there's no Y. Okay, so any of them that have the letter Y in it are the pyrimidines. That's an easy way to tell. And only certain bases can bond together. So, this is the way it would maybe look in our model, maybe. Here we go. So here's a nucleotide, phosphate sugar base, and let's say we had another one. Well, if this was an A, then the base that would stick to it, we'd have to flip this upside down for the bases to be together. And if that was an A, then this would be a T. Now, I'm not going to write the T upside down just because that would be a little confusing. Okay? And let's take another one. Well, here's another nucleotide. And let's get another one. And we're going to have to flip this one upside down too. And let's say this one's a letter C, so that would make that the letter G. C's and G's go together, A's and T's go together, and we could repeat this multiple times, over and 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 over. And the only thing that's changing every time is what sequence this base pair is. So these, this sugar would connect to the phosphate, sugar, phosphate, and it kind of does this on the sides. And what sticks out in the middle are these partners. So if I had T and A here, the other side would be, and it just keep going. Okay, so that's why a lot of people didn't pay attention to DNA, is because it's really only 6 different chemicals. You've got sugar, you've got phosphate, you've got adenine, thymine, cytosine, and guanine, but the order of those bases is what makes it important. Hopefully this helps you understand the basic structure of DNA a little bit.